Doctor, thanks for being with us today.
Thanks for having me, Dave.
Let's start with a brief overview of your technology today, and then talk specifically about the COVID-19 test that you're developing, particularly as it relates to the polygenic risk score in genomics.
What we do is risk stratification, and what that means is we identify people who are at high risk, average, and low risk of developing a disease. Our core technology is applied to what we refer to as common complex diseases like breast cancer, colorectal cancer, type 2 diabetes, and heart disease, just to mention a few. And by identifying high-risk individuals, we are able to use all our resources more effectively by targeting these people at risk. And at the same time, we can reassure people who are at low risk. And as such, we create a more personalized, precise approach to these common complex diseases, which are responsible for the majority of deaths and disease. In the same way, we can use this platform technology to assist with the COVID-19 pandemic. And what it does is it identifies people who have natural immunity and natural ability not to have serious complications from this disease. By identifying these resistant groups, we can then make sure that the first responders, whether they be medical, policing, government, are safe and less inclined to develop life-threatening complications. At the same time, we can isolate and practice preventative health in people who are at high risk. So as a result, instead of isolating the whole population, you simply isolate the people who are deemed to be vulnerable, and in the meantime, the secure or the immune group or the resistant group are able to continue practicing their craft. So it means that we can quickly return back to normality and not risk our vulnerable populations.
Let's talk about, again, your technology and the test that you have today and the polygenic risk score.
So the polygenic risk score is a genomic platform. It's a concept that started roughly 15 years ago. And what it does is it assigns a value to your risk of developing a disease. A polygenic risk score of 1 equates to a risk of average, an average risk. A polygenic risk score of, let's say, 2 is double the risk, etc., etc. And this risk score can be applied not just to complex diseases, but to infectious diseases such as the COVID pandemic. And what it does is it allows us to identify the people who need our protection the most and the people who can carry on without the need for special requirements. It's a platform that can be applied to oncology, which is cancerous diseases, metabolic diseases such as diabetes, etc. It is a safe, inexpensive, and accurate way of identifying these types of individuals. And it's a science that's really only emerged over the last 10 years, with the tipping point being last year. So this is an ideal environment for us to really show how modern medicine can help society in these difficult times.
And tell us more about your CLIA labs and your ability to get to market quickly with this new COVID-19 test.
Yeah, so we've been doing this type of work for quite a while, and we're uniquely placed that we have a CLIA, which is the American certification, and NATA, which is the Australian certification. This is a unique certification, certainly where we are based, which is in the Southern Hemisphere in Australia. And it allows us to service both continents effectively and inexpensively. This also allows us to develop new tests within a very short period. So we can turn a new test around now in 3 to 6 months.
And what would be the cost of developing this test?
Well, we've got a team of 6 working here, and we collaborate with a number of institutes around the world. Most tests would cost us somewhere between $1 and $1.5 million to develop. And as I mentioned, it's a 3 to 6 month exercise. We usually require 3,000 to 6,000 samples before we have a reasonable amount of data to give us confidence in presenting this type of test to the community. I'd like to add that the more data we have, the better these tests become. And the flexibility that lab-derived tests allow us is to continuously upgrade our algorithm and be relevant as more information becomes apparent.
To summarize, let's talk about how this test will be different than other tests currently on the market.
This test allows us to use your immune system to develop a strategy or an outcome. So what we're looking at, when applied properly, this test means that we'll create immunity without immunizing, simply by exposing people who are unlikely to have serious complications to the infection. As a result, we could quickly return back to a normal, safe environment, as opposed to the old system, which would be to develop a vaccine, scale up manufacturing, then implement the vaccine through a medical process. These are all time-consuming and expensive exercises. This would occur in parallel, of course, but it would mean that, again, the people requiring immunization would be targeted. The people requiring isolation would be identified. And as a result, we would have a more efficient and more streamlined and less expensive approach to the whole exercise of immunizing the community. And it also means that there would be less complications, less morbidity, less death from the vaccine itself.
So you'll develop a genetic risk score for individuals by examining people that have already had the disease, basically.
Correct. Yeah. The key to our technology is we study the DNA of people who've had the disease and had the worst version of the disease with the DNA of people who've had the least invasive version of the disease. So now we have the strengths and the weaknesses of the immune system in dealing with this type of a pandemic. And as a result, we can identify these individuals and assign tasks to them which do not put them or the community at risk.
Fascinating. Thanks for being with us today.
Thanks for having me, Dave.